Good afternoon. Good afternoon, ladies and gentlemen. Let me welcome you at today's Mendel's lecture, which will be given by Professor Manolis Kellis from MIT and Harvard University. I will introduce today's speaker briefly. He received master's from MIT, Massachusetts Institute of Technology in 2000. 3 years later, he defended his PhD from the same institution. Then he undertook a research stay focused on yeast genetics in called Spring Harbor Laboratory. And in 2004, he undertook a postdoctoral stay on evolutionary genetics at MIT and Harvard. Then in 2004, he became an assistant professor, 2008, associate professor, and 2014, full professor at the Department of Electric Engineering and Computational Science at MIT. His research interests are focused on gene regulatory maps and circuits, and he's trying to understand how these circuits relates to human diseases. He was involved in a number of very important projects, like a NIH Roadmap Epigenetics project and Encyclopedia of DNA Elements ENCODE project. These are very large projects focused on sequencing and then analyzing genetic information. He published 208 publications, so ordinary number, but the number of citations is remarkable, 35,000 citations for these 200 papers. He received a number of awards. I will name just 3 of them. MIT Award for Best PhD in Computer Science. Then he was elected as one of the top 30 principal investigators of the future in genomics. This was by Genome Technology Medicine. And one of the most prestigious awards which he received was the US Presidential Early Career Award in Science and Engineering. And we are very much looking forward to your presentation, Manlio. The floor is yours.
This is wonderful. Oh. Hello, testing. All right. Wow, it can be done. So as I was saying, it's an honor to be sitting here in the place where Gregor Mendel founded the field of genetics that we rely so much upon today. And what I'm going to tell you about today is my own personal journey in using Mendel's legacy and what he has left us with, and what we have built upon in order to actually understand human disease and sort of make a difference for everyone here today and for our children and our grandchildren in the future. I mean, we live in a very privileged time, and I hope you guys realize that. Humanity has never been in such an incredible place scientifically. We understand the universe from, the quarks all the way to the black holes. We understand biology from, the gene and all the way to the systems level. We understand math and physics and, chemistry at a level that no other generation has understood. And to me, it is a privilege for all of us to be living through this time of discovery. Humans have never lived for so long. World hunger has never been at such a low level. I mean, this is incredible that a child born today has a life expectancy that has never been at this level. And a lot of that is thanks to Mendel. A lot of that is thanks to those peas experiments, these crossings that happen in this very place. So I hope you guys realize the importance of this edifice, and also just how fortunate we are to be living at a time like today. So as this is a historical lecture going back to Mendel, and as I'm Greek, I thought I would go back a little further than that. So what I'd like to tell you about is the story of inheritance and our understanding of genetics in prehistoric times. And it is no surprise to anyone here that humans have understood heritability and inheritance since the dawn of time. If you look at selective breeding of animals, of crops, I mean, which one would you rather eat, this or that? This is teosante, and this is modern corn. Genetic engineering, which took thousands of years. Which one would you rather have a dinner with? This one? Or would you rather bid dinner for that one? This is basically the same species except for domestication. So humans realized from the dawn of ages that if you take the individuals that have the phenotypes that you like the most and you make them breed, you will get something that will continue resembling that phenotype. Humans also understood that kids looked like their parents, and that traits that are almost Mendelian, like oligogenic traits, like hair color and eye color, etc., were in fact inherited from parent to offspring. This has never been a problem. The problem was, what is the basis for that heredity? What are the rules? And that was the revolution that Mendel undertook. But being Greek, I can't help but give a little credit to some of the folks that preceded Mendel. So around 550 BC, Anaximander, along with many, many mistakes that he made, reasoned that the first human must have been born from some non-human relative. I mean, this is an idea that you would have probably gotten hanged for in 1200 AD, 2,000 years later. But these are revolutionary ideas. He had an idea that there was a fish origin of land animals, that somehow the land animals came out of the sea. Aristotle, in 300 BC, built a taxonomy of life that lasted for 2,000 years. It was not surpassed for another 2,000 years. It became the reference. After actually traveling for 10 years in the island of Lesbos that my father actually grew up in, he basically studied the natural life there and sort of built, characterized 300 plus species and dissected dozens of species and, studied anatomy and so on and so forth. And theories about inheritance, about how the male seed and the female seed get together and make an offspring, have been attempted for hundreds of years by Theophrastus, Hippocrates, Aeschylus. So all of these scientists certainly made many mistakes, but they also had ideas that were revolutionary even thousands of years later. And if you look at the concept of natural selection, of random mutation followed by natural selection, that also existed in ancient times. Empedocles, picture here, and Epicurus, picture here, had already concepts of random mixing of traits, of natural variation, out of which the successful ones survive. And very clearly said that can give the semblance of purpose. And that's extremely important, the fact that we as humans tend to ascribe purpose to a lot of things that we see. We say, oh, this is designed for that, and this is, designed for this. But no, the process of evolution is something that comes from this random mutation generating a mutational pool, and then brutal selection, selecting the fittest in a completely, unbiased way. And then Epicurus basically made the transition to a purely naturalistic generation of this diversity without any supernatural intervention. So that was, again, quite groundbreaking at the time. But you have to also realize that at the same time that these extremely forward-looking philosophers and scientists and naturalists were making these theories, there was, of course, another school of thought, Plato and the Stoics, that basically thought about unmutability of life. And I think that's something that Darwin had to really break against, because those ideas of the Stoics were in fact embraced by religion and by Christianity and became the dogma, at least in the Western civilization, for again, more than 1,000 years. So this is the backdrop against which we are now entering the Renaissance and this dramatic transformation of this field of understanding inheritance and evolution. The dominant idea in the century of Mendel was in fact proposed by Lamarck. And the whole concept of Lamarck was this transmutation, the concept that a species would progressively change, these very small progressive changes. And that was very revolutionary compared to the dogma of everything is immutable and God on the sixth day, by the sixth day had created all of the orders of life. And these were the orders we have. Lamarck's ideas were actually quite revolutionary by basically saying that, there's this natural fluid transformation of species. But of course, he was extremely wrong in other ways. He basically saw this progression from, the lifeless to the full of life kind of thing, just like the ancients did, of increasing orders of complexity. And he thought that there was one life force pushing towards complexity, and then another life force creating this diversity, so that there was this spontaneous generation of different life forms. That, the image that is usually shared is the giraffe stretching its neck, and on the next generation, the giraffes have taller necks. And that was very much the dogma at the time. Darwin didn't actually fight against that dogma of Lamarck. It's kind of interesting. What Darwin's contribution was again this concept of the continuum of species, this concept of random mutation, which generates diversity, and then natural selection that generates fitness. So rather than a directional evolution, it was very much a random path out of which different niches would be occupied. So it's no longer, hey, we're tending towards perfection. It's more that different niches are created through changing, geology and changing biology and changing environment, and then these niches are occupied by this generation of random mutation and then selection of that. But Darwin was also very wrong. He was basically missing what, ironically, Mendel had published just 1 year earlier, which was the laws of inheritance. So Darwin had basically this concept of what we call today Lamarckian evolution. He had basically this concept of pangenesis, where the body would basically be sending signals called gemmules to the sperm and egg, and then these would then later develop to the embryo. And this is fundamentally wrong, and that is in fact what led to Darwin's theory being pushed aside for a good 50 years before it was reconciled in the modern synthesis of evolutionary thinking. So, and that's the tragedy of Mendel. He published his paper in 1866. He gave his first lectures in 1865. His paper was cited 3 times in 35 years. If only that paper had made a bigger impact, we would have had genetics, probably a century earlier than we did. So, but again, what was dramatic about Darwin was the fact that he took something discrete and he made it continuous. Instead of sort of having every species occupy a completely separate spot, he basically said, no, there's a continuum of species, and this is sort of where diversity comes from. Mendel had exactly the opposite. So Mendel, his big contribution, in my view, was this whole concept that instead of sort of having this blending inheritance, which was the concept at the time that almost every scientist had, and that was fundamentally flawed, he basically came up with particulate inheritance, the concept of units that are not diluted away, units that are discrete. And the dominant and recessive law, his third law of genetics, in fact, explain how blue-eye color could jump a generation or jump 10 generations. The fact that recessive traits could persist unchanged, not diluted away. And I think that in my view, is one of the biggest changes in thinking. This whole concept of particulate inheritance, no blending, really discrete units of inheritance, which were later named genes, and then discrete versions of these units, which were later named alleles, that were either dominant or recessive. And of course, his law of independent assortment, that every single one of these traits was inherited independently. And again, it turns out that he was, almost right. Basically, he had chosen 7 traits. He had been extremely careful to basically have true breeding parents that would be only white every generation, or only purple every generation by doing self-crosses. And then, starting from that material, he selected 7 different traits that were independently inherited. And it turns out that 2 of them were kind of on the same chromosome, and maybe there's some theories about whether he fudged the data or not to make it really look like independent assortment. But what he did was revolutionary. I mean, what's really remarkable is that Darwin, at the same time, had done the same experiments. He had crossed peas, and he had found that they could jump a generation, and he had no idea how to interpret it. And I challenge you all to go through the garden and to look at these diagrams. And sure, the first one that I'm showing here is kind of simple, recessive, and then when you have both recessive together, kind of like blue eyes, you end up with blue eyes. But if you have even one brown-eyed allele, you end up with brown eyes because it's dominant. But then, that's because we know genetics now. Picture yourself in the shoes of someone who has no idea what chromosomes are, no idea what DNA is, no idea that there is a germline, and yet coming up with this absolutely beautiful model. And that, in my view, is what genetics draws its strength from. Genetics works regardless of what the physical instantiation of that inheritance is. And that's, I think, the power of genetics even today. When we're using genetics in our own work, we rely on the fact that it doesn't matter what the mechanism is. I can do genetic mapping. I know that something is here that is influencing this phenotype, regardless of what place in the body this thing might be acting, regardless of when or where or how. Whether it acts, maybe the first month of gestation, I will never know. But I know that something is there that is influencing phenotype. And I know that something is discrete. And as a computer scientist, I'm very fond of that. To me, this is the first digital revolution, if you wish. The fact that we went from continuous blending inheritance, which would absolutely not work for evolution. Evolution just simply would not work because you have a new advantageous trait. Every time you cross with another individual, you blend that out and you dilute it out, and then it gets lost. There's no way of evolving new life forms. Everything would just blend towards the middle without this concept of digital, of discrete inheritance. And I always like to say to my class that humans like to boast that we invented the first digital computer, whereas in fact, we are the descendants of the first digital computer. Inside every one of our cells lies a digital code, and that's why inheritance works. Because that digital code enables undilutable material, which is replicated perfectly. This does not exist anywhere else in the world. If you look at physics, there's no law of physics of perfect preservation of something, generation after generation after generation. That's where biology defies physics. This is the very principle that Mendel discovered that completely changed the view of our understanding of life. But Mendel was not accepted. Even himself, he doubted his own experiments. he basically could not reconcile why things worked so beautifully for his peas in this very garden, and why these rules would ever apply to complex traits like height or skin color or, all of these different multigenic traits that we observe every day. So just like he could not reconcile that, he had actually done experiments on other species that did not follow the same rules of inheritance. So in a way, his ideas were dismissed as this esoteric thing that happens in peas without actually recognizing that the same laws govern this huge diversity of quantitative traits that, everyday life is all about. And that's where the problem of biometrics, this biometric school versus the Mendelian school, basically could just not come to terms with each other. And biometrics was clearly observing continuous phenotypic variation. If you look at hair or eyes, it's not just blue or brown. It's, all of these different colors because there are multiple genes and multiple alleles for each of these genes, in contrast to Mendel's very beautiful, very clean experiments. And that basically was a huge problem. People had a big, big trouble embracing Mendel even after the rediscovery of Mendel's work and Mendel's papers and Mendel's laws. So even after 1905, when three different authors, 1902, when three different authors independently rediscovered Mendel, the biometrician school was just not able to come to grips with that. And that's where we come to the 20th century, where Fisher, who's pictured here, wrote this seminal paper in 1918 that completely transformed the field. He was not the first to say this. In fact, in 1902, I believe, there was another set of authors that had already sort of made that statement, but the ability to change people's views is part of what made the recognition for Fisher, because this other paper had just simply not been as well embraced by the scientific community. And that's also where Mendel's legacy is so important, because a lot of people talk about the rediscovery of Mendel's law. How? By basically people doing experiments and then independently coming up with the same conclusions. But there's a lot of controversy around that. In fact, one of the three authors is thought to never have even understood Mendel's laws. And then the other two, it's unclear whether they were able to interpret their own experiments without Mendel's insights. So I think the legacy of Mendel comes from being decades ahead of his time in being able to interpret these extremely complex experiments and come up with this set of three very simple rules. So what did Fisher do? Basically what Fisher did is reconcile this continuous variation in phenotypes that is observed every day, and this extremely discrete, quantile, particulate inheritance of individual units of genes. How? By basically saying, here's a very simple mathematical model of polygenicity, where there are multiple Mendelian loci that are all inherited independently, each with an additive effect on whatever phenotype you care about. And if you look at that, it only takes 5 or 6 loci to basically end up with, continuous variation or seemingly continuous variation. And that finally reconciled this fight between the Mendelians and the biometricians. Another revolution was by actually understanding that there was a physical medium that embodied this genetic material that was passed on. It wasn't just some fictional piece of information, it had a physical nature. And that's where the chromosomal theory of inheritance and eventually the solving of, the understanding of DNA as the genetic material, and ultimately the solving of the DNA structure in 1953, thanks to the X-rays by Rosalind Franklin and the modeling by Francis Crick and Jim Watson, that basically showed that this incredibly beautiful structure of this double helix is not just beautiful, but it explains heredity extremely simply. I have a 6-year-old son, a 4-year-old girl, and a 2-year-old girl, And I had my son draw me the second strand of DNA when I drew the first strand of DNA. My 6-year-old was doing, replication. It is that simple. Basically, as soon as you have one side, you can immediately infer the other side. And that made a very famous, it does not escape our notice that this model immediately implies a mechanism for inheritance, which will be covered elsewhere. Then, again, going back to Mendel, and this little discrepancy from his numbers of 9:3 to 3:1 ratios that you would expect in his P's, genetic linkage was born by understanding that this deviation from Mendel's laws was in fact due to the proximity of different genes in the same chromosome. And that was fundamental in what is now known as Mendelian gene mapping. By basically being able to see how a particular trait segregates in relationship with markers along the DNA, we were able to now start mapping a huge number of diseases like Huntington's to individual loci and ultimately individual genes. So that was the big revolution of the 80s, the fact that we could now start mapping human genes thanks to these laws of inheritance. And here we are today, in the 2000s, if you wish, where we have now achieved what seemed unthinkable. We have not only understood the laws of heredity, thanks to Mendel, we have not only understood the embodiment of the physical material that carries out this inheritance, we have gone ahead and actually sequenced the entire human genome. It cost $3 billion, but we did it. This is ridiculous that a species could come together and sequence its own genome. I mean, that's the ultimate level of introspection. Not only did we do that, but we have also built maps of all the common variation across humans. Anything that's commonly inherited that was there in the 10,000 people that came out of Africa has been mapped. And now for every major population, we also know all of the common variants. This is basically a complete catalog of common genetic variation. We have built haplotypes and used them to recognize that genetic segments are inherited in groups. And therefore, we can build a genotyping array and only map a few, hundreds of thousands of variants in a single experiment that cost, a few $1,000 to basically now genotype hundreds of thousands of individuals. And that's where genetics comes in. Because we're able to look across human chromosomes and map all of the genetic variants that are segregating along with a particular phenotype, we're able to say that this particular place in the genome plays a role in that phenotype. And that's where the beauty of genetics comes in. It does not matter how this is encoded in the genome. It doesn't matter what cell type it acts in. It doesn't matter what tissue it acts in, when it acts in, and so on and so forth. Genetics simply tells you there's something here that is extremely important. And we are now, with the dramatic reduction in sequencing costs, able to get at the rare variants. And that's finally coming full circle back to the Mendelian inheritance of strong phenotypes. Why? Because common variants, the reason they're common is because they have weak effects. Otherwise, evolution would just not have tolerated them to be so common. If something has a strong effect on schizophrenia, and then everybody who has it has schizophrenia, their number of offspring is simply going to go down. And you're not going to see that variant at high frequency anymore. Selection will work against the high frequency of that variant. So basically, strong effect variants are maintained at low frequency in the human population, whereas weak effect variants are allowed to rise to high frequency. So that basically creates this dichotomy between common variants that are having very, very weak effects and rare variants that are allowed to have much, much stronger effects. So this is the picture that we get for every one of these dots in the previous slide. So this chromosome here is down there, and that's chromosome 1, 2, 3, all the way down, and then every single one of those dots is a different type of trait that has been mapped to that location in the chromosome. When I first started showing this slide, there were a few dozen. And now, like this is actually today. I did this like an hour ago. It's thousands upon thousands of variants. So every single one of these dots is a picture like this. It basically says that if I take all genetic variants that are associated, all genetic variants in the genome, and I ask which ones of those are biased to be more frequent in individuals that have a particular phenotype, then I end up with something that we call a Manhattan plot. Why do we call it a Manhattan plot? Because it looks like big skyscrapers, okay? So this big skyscraper here is the FTO locus. It stands for Fat and Obesity Associated. It was renamed to Fat and Obesity Associated after it was discovered completely unexpectedly that this locus held the strongest genetic association with obesity. If you ask anybody in the street today, why is this person heavier than that other person? The answer is because they eat more and because they exercise less, right? So there's this stigma of obesity is your choice. And back in 2007, when the first genetic association with obesity was discovered, we suddenly realized that there's a genetic basis to obesity. That yes, you might eat the same amount, but you're going to gain more weight. And you might exercise the same amount, but you're going to gain more weight. So that basically led to this dramatic infusement of funds to try to figure out how this gene works. And if you look at this particular region of the DNA, I'm now zooming in, and I'm showing every genetic variant, which is common in the human population, and how it is associated with body mass index, a quantified measure of obesity. And you can see that there are 89 common variants here that co-segregate with a trait, and they're all sitting right in the middle of this FTO gene. Nowadays, pharmaceutical scientists in industry are basically saying, oh, bummer, there are 5 genes in this genetic region. It's going to be really hard to figure out which one is the true target. In this particular case, everybody felt they hit the jackpot because there was only one gene. So they thought they were done, and they went after FTO. It turns out that it's an RNA methyltransferase, and suddenly epitranscriptomics became a big thing, and everybody started working on that because it's probably the basis of obesity, and so on and so forth. I'm going to make a long story short to basically tell you that the FTO gene that sits exactly there with no other gene in that region has nothing to do with obesity. So here I am telling you that number 1, genetics works, and number 2, FTO has nothing to do with obesity. How do you reconcile the two statements? You reconcile the two statements by basically asking what fraction of the human genome codes for protein. And the answer is about 1.5%. About 99% of the human genome does not encode proteins. And if you look at what portions of this very, very long gene encode proteins, you see a tiny little dot here called exon 2, a tiny little dot here called exon 3. None of these genetic variants overlaps with a protein coding position. None of them perturbs the protein of FTO. Therefore, it must be some kind of gene regulatory function. It must be something that controls the activity of genes, but not the protein itself. And this is not just some esoteric, scholastic, academic exercise. I'm not saying, oh, wow, we found an exception. This is true for 93% of these common variants. Remember, common variants have generally small effects. And if you look genome-wide, these small effects come from generally perturbing the control circuitry of these genes rather than the protein of these genes. Who's with me so far? Raise your hands if you are. Awesome. Great. Fabulous. So if it's a control circuit, if it's a control diagram that we need to understand, we have to figure out the wiring. What we know is that a switch on the wall broke, but we now have to trace the wires to figure out where is the light bulb that lights up when I flip that switch. And when we traced the wires in a paper that we published a couple of years ago in the New England Journal of Medicine, we basically found that this switch here flips a light bulb that's sitting 2 buildings away at this resolution. 1.2 million nucleotides away. There's a good, few genes between that switch that sits in the middle of this gene and the light bulb that it turns on and off. I was introduced at a symposium recently as the guy who wrote the most depressing paper of 2015. Because all of pharma was so happy when there was a single gene there, and they were going after this. And now they're like, oopsie, we may be going after the wrong thing. So what I'm going to tell you today is how we can use all of these modern technologies to deliver on the promise of Mendel, to deliver on the promise of genetics, and to basically understand what are truly the targets of all of this genetic variation that sits in the human genome, that is underlying thousands of associations that have already been discovered for so many different diseases. Why is this important? This is important because genetics works. This is important because when I see a genetic difference, it is unbiased, it is uncorrected, and it is causal. What does that mean? That means that it's not because I was studying this pathway that I ran into this gene. I didn't go after a few candidate genes. I didn't just perturb a gene and then notice the phenotype in mouse. This is a place where even a minute change in the gene regulatory circuitry of this gene, in the control of this gene, has a statistically significant, undeniable effect on the phenotype of that disease. That basically means that if I find a way to modulate this gene, by golly, I will have an effect on that phenotype. And if nature allows that gene to be modulated by 2%, and I go in pharmaceutically and I modulate it by 50%, I'm going to have an effect that's 25-fold stronger than the natural genetic variation. So common genetic variants are your friends. They're not your enemy. Many people say, why do you care about this? It has a 1% effect. But I care about this because it has a 1% effect on the phenotype, not on the gene nearby. If I have a very small effect on the phenotype and a very small effect on the gene nearby, if I increase the effect on that gene, then the effect on the phenotype can actually increase. So pharmaceutically modulating a strong effect variant that completely knocks out the gene and has a very small effect on the phenotype, that's the best I can possibly do. I've knocked out the gene completely already from genetics. That probably means that that gene doesn't matter that much. But if I have only a tiny little effect on the gene and it has a noticeable effect on the phenotype, that means that pharmaceutically I can actually get a much bigger effect. Okay? Who's with me so far? Cool. So that's why genetics is so cool, because it's unbiased, it is causal, and it is uncorrected. That basically means that all of the feedback mechanisms that have made countless drugs fail are not acting there. They're simply not correcting for that. Basically, that we have an entryway into modulating a phenotype that is not naturally corrected by the body and therefore erasing its effect. So that's the promise. The promise is that we'll be able to find new disease mechanisms that are completely unexpected. We're going to be able to find new target genes. We're going to be able to develop new therapeutics and ultimately enable personalized medicine. But the challenge is understanding the mechanism. Again, we saw throughout history, you can have a model, but if you don't have the mechanism, there's not much you can do with that model. So what do we need to know? We need to know the target gene. We need to know what is the causal variant. Because with common genetic variants, they're co-inherited. All of these guys are always there or always not there. If I have the, version 1 here, I'm going to have version 1 for all of these guys. And if I have version 2 here, I'm going to have version 2 for all of these guys. So it's very difficult to find which ones of those is the causal one. So that's challenge number 2. Challenge number 3 is we don't actually know what cell type this acts in. Great. I've now told you that it's a gene regulatory variant. Where does it act? Does it act in the brain? Does it control my appetite? Does it act in the brain to control whether I like to exercise or not? Does it act in my muscle to make it easier for me to exercise and less painful? It could be acting anywhere. And it could be acting on any pathway. And most importantly, the target gene could be far, far away. So what we are doing in my lab is trying to sort of go that last step of understanding the mechanism. So we're starting with genetics. We're starting with common and rare variants, and then we measure the effect of those variants at the molecular level. Again, we're so privileged to be living in this time when all of that is already elucidated, so we can just go and figure out the expression of all of these genes in hundreds of different tissues and cell types. We can go and measure the epigenome. So the genome, we all know and love. It's this double, helix. It's absolutely gorgeous. But the genome doesn't float around naked in the cell. It is packaged very minutely around nucleosomes. And every nucleosome has about 147 nucleotides surrounding it, and another, 40-some nucleotides of a gap. So you can think of this as 200 base pair packets of information. But that packaging is not just structural. It doesn't just hold it together. It's also informational. It's as if you had neatly packaged all the rope in your house, but also labeled the boxes with, here's an enhancer in orange, and here's a transcribe region in green, and here's a promoter in red, and here's a repressed region in gray, and so on and so forth. So all of the cells in our body have exactly the same information. Everything that it takes to make a human is encoded in every single cell, almost every single cell of our body. That information comes in a massive encyclopedia called the Human Genome. It's 23 volumes, 23 chromosome pairs. And from all of that information, if you take the analogy of different professions, you have, imagine all of human knowledge in 23 volumes. And if you want to be a physicist, you read different chapters or different passages. And if you want to be an artist, you read other passages. If you want to be a geneticist, you read other passages, and so on and so forth. All of human knowledge, and you get to choose, depending on your profession, which part you want to read. So that's what happens in our cells. All the cells have all of that massive amount of information, but they can't deal with it all. If I'm a plumber, I don't need to know what a nuclear physicist does, and if I'm a nuclear physicist, I don't need to know what a mathematician does. It's just different professions need to know different things. How does the cell do that? By basically coloring these packages, by sort of adding, bookmarks on this book of life. And these bookmarks are like Post-it notes, or underlining, or highlighting, and so on and so forth. And all of these different marks exist in different modalities. So methylation of DNA is usually a mark of repression. That basically means that in this cell type, I probably don't need this region. DNA accessibility is a mark of usefulness. Basically, the stuff that you care about, you keep it accessible. You don't package it tightly. And then histone modifications, there are hundreds of them. So we can encode a large number of different types of functions. You basically can say, this is an enhancer, this is a promoter, this is a transcribe region, this is a repressed region, this is polycomb repressed, and heterochromatin, and so on and so forth. And all of these different types of epigenomic function are encoded in this code. So what we do is basically we start with genetics, and then we map the activity of different tissues and cell types systematically. And then we employ computational tools to integrate all that information and to discover patterns. This is a big data approach. This is not a hypothesis-driven one by one. This is basically saying gather all the data and figure things out later. Okay? And that's the approach that Darwin took. That's the approach that Aristotle took. That's the there's two schools of science. One is the, test a single hypothesis, which is what a lot of physics is about nowadays. You basically gather massive amounts of information, and in the end, you have one bit of yes, no with a significance. Whereas in genomics, you gather massive amounts of information, and you can reuse it for all kinds of new questions because you're looking for patterns, and from these patterns come hypotheses, which you can then test using new types of experiments and new data gathering. So anyway, we basically gather all that data, and we predict what are the driver genes, what are the driver regions, and what are the cell types underlying all this. And then lastly, once we have made these predictions, we actually go in and test them. We go and manipulate these genes in cell cultures or in primary cells or in mouse models, and we basically then disseminate these results to the scientific community for everyone to use, including our competitors, including pharma, including people who want to make money from it, everyone. Because that's our goal as scientists, just create knowledge and disseminate it as widely as possible. Okay? So we basically transform this information from saying, hey, there's something going on here based on genetics, but I have no idea what it is, to this is the circuitry of that region, and these are the mechanisms that may be explaining this association. Okay? So genetics has both a blessing and a curse. The blessing of genetics is that you will find that association regardless of how it acts. Ed Lewis used to say, even if genes were made out of green cheese, genetics would still work. And it doesn't matter what the underlying mechanism is, genetics will basically say this is the locus that matters, this is the region, there's something going on here. The curse is that, yes, you found the locus, but it could take another 10 years before you figure out that the target gene is actually 1 million nucleotides away. Okay? So we basically address the curse part of genetics by basically taking all of the genetic variants, figuring out what control elements are they under by using this epigenomic code. We basically can use these signatures that I showed you on the previous slide to denote different classes of regulatory elements. So promoters are regulatory elements that are proximal to the gene, and then they guide where transcription starts. Enhancers are regulatory elements that are far from the gene, and they guide when that gene will be expressed across different tissues or conditions and so on and so forth. So the where and the when we can use to basically say, aha, this SNP here, this single nucleotide polymorphism, overlaps an enhancer region, a control region, which is active in those cell types and is bound by this transcription factor based on this regulatory motif, and it targets this target control gene. Okay? Who's with me so far? Yay. All right, so now let's see how that circuitry changed this FTO story. So for 10 years, scientists invested $1 billion to understand that locus and to understand the function of that gene. Nothing came out of it. Then we basically said, okay, let's bring all of that massive data and apply it to that single locus and see what does the information tell us. And what we found is that there's a 12,000 nucleotide long enhancer. This is a massive enhancer. I mean, super enhancers are defined are things bigger than 200 nucleotides. Sorry, bigger than 2,000 nucleotides. This is 12,000 nucleotides. This is dramatically big. It's this massive control switch. Instead of like tiny switch on the wall, this is a panel. This is like a big, massive thing. And that panel is not active in brain, as many people had suspected. Many people said, okay, this locus is probably acting to control your appetite and your exercise. If you take basic, thermodynamics, basically there's energy in, energy out, right? So basically, how much do I eat and how much exercise do I do, right? Everybody with me on thermodynamics? Am I missing something? Of course I'm missing something. Energy lost. So if that big panel on the wall is heating up, that's energy lost. That's something that we don't intuitively think about when we think about metabolism. We think energy in, energy out. How much do I eat? How much do I expense? But there's a third component of heat. And turns out that heat dissipation is something regulated by our cells. Our fat cells can be our best friend, and they can also be our worst enemy. In times of starvation, when food is scarce, fat cells store store calories in high energy molecules known as lipids inside white fat cells, and that's what makes us chubby. So basically, when you're chubby, it's because we evolved in times of food scarcity to store extra food in our diet, extra food in our daily intake that we did not burn, to store it as energy. But this is a controlled process. For some of us, our fat cells can choose to instead burn these excess calories. How? By depolarizing the mitochondrial membrane. The way that energy gets created in cells, the way that life fights against physics, is by creating gradients and then fighting against the forces of entropy. And these gradients are in fact enabling you to store energy. To lose energy, all you need to do is just let go of that gradient. And that's what our fat cells can do. They can just simply burn calories by depolarizing that gradient, that mitochondrial membrane, getting rid of that gradient, and then dissipating energy as heat. Okay? That's called, another Greek word, thermogenesis, generation of heat. Okay? So when a fat cell develops and differentiates, it basically chooses its profession. Am I going to be an energy burner, or am I going to be an energy storer? And in this FTO locus lies this massive control switch on the wall that basically decides whether you're going to store energy or burn energy. IRX3 and RX5 are the two light bulbs at the end of that massive control panel, a million nucleotides away, that basically control whether you're going to turn on these genes and turn on thermogenesis, or whether you're going to turn on these genes and turn on fat storage, energy storage in your fat. Okay? Everybody with me with that part? So now, what happens when you have that genetic variant? When you change this AT-rich motif, AAT, which is bound by this very creatively named AT-rich interacting domain, ARID5B, when that protein can no longer bind because you've now changed the T into the C because of that RS1421085 genetic variant, which is common in the population. This genetic variant is at this allele here, the T allele, is in 42% of European chromosomes, including both of mine. So mom and dad, thank you. They gave me 2 risk alleles. Everybody's looking at my tummy now, I'm sorry. So they gave me 2 risk alleles for this FTO locus. My wife has none. So, you know, you can guess when we go to the beach which one has to cover up. All right. Oops. 42% of European chromosomes carry the risk allele for obesity. 44% of Southeast Asian chromosomes carry the risk allele for obesity. What happens, and you guys should be concerned, is that when I have the T here, when I have the T here change into a C, I no longer can bind. ARID5B is a potent repressor that goes on that control board and turns off these RX3 and RX5 genes. And when that nucleotide is no longer there, that switch no longer functions, and it's stuck in the on position. Now the question is, what does RX3 and RX5 do? What they do is they normally, when they're on, repress thermogenesis and turn on lipogenesis. When they're off, they let thermogenesis happen, and then they don't turn on lipogenesis. So that basically means that for those individuals who carry the risk allele, they're simply unable to burn calories. They store them all. And that basically results in 7 extra pounds for the homozygous risk individuals.
Why is this exciting? This is exciting because we started from a completely unbiased view of the genome. We just looked at where are, all of the genetic variants that are associated with obesity. We found FTO as the strongest genetic variant associated with obesity. Then we basically said in a completely unbiased way, let's profile all of these different cell types to see where it's active. And we found that this region was active in pre-adipocytes during the first 3 days of differentiation. As these pre-adipocytes differentiate, they basically decide their profession. And if that switch is stuck on the on position, all of them are basically going to start storing fat, storing calories. And then, with the same exact diet and exercise, some of us will gain weight, others will not. Okay? But the beauty of figuring out this is not just that we can go from a genetic finding to an insight. It's that we can go from an insight into a therapeutic. How? Well, we have the whole circuitry laid out. Oh yeah, I think it's okay to turn them on. This is very unusual. Most of the time when people sit on the wall, they turn off the lights. So please, use your ARID5B to turn on the lights. Whoever did that. Okay, no lights. All right, so the beauty of figuring out these switches on the wall is that we can now go and flip them, okay? We have the whole circuitry. Notice, however, that the FTO gene is nowhere to be found. Who cares about the FTO gene? It's just that locus that was called FTO. We can now start intervening. We can basically use molecular techniques of overexpression or knockdown or knockout of these genes to turn on or turn off the upstream regulator, to turn off or turn on the downstream target genes. And we can even use something called CRISPR-Cas9. You guys have heard of that? Yay. To basically edit the genome. We can basically take primary adipocytes from obese individuals who have the risk haplotype and the risk genotype and basically edit the genome of their cells to change the C back to a T and restore this binding site for that protein. How do you think these experiments worked out? The answer is ridiculously well. This is editing of a single nucleotide out of 3.2 billion nucleotides in the human genome. I mean, when it comes to precision medicine, I don't know how much more precise it gets than 1 in 3.2 billion. By editing that single nucleotide in primary adipocytes from risk individuals and changing that C into a T, we were able to fully restore the process of thermogenesis. These cells are now perfectly fine. They're able to, burn calories. What about the organismal phenotype? We basically created a dominant negative IRX3 mouse. We basically took normal mice and we created a construct that expresses a dominant negative form of IRX3 in adipose tissue. These mice lost 50% of their body weight. They're here. As they grow, they just are much smaller to start with. And then, when you put them in a high-fat diet, the normal mice gain weight. These mice are unable to gain weight. They just simply burn the calories. The switch is now stuck in the thermogenic position, okay? These mice did not change their appetite. These mice did not change their exercise. These mice just simply burned more calories when they were awake at night and when they were sleeping during the day. Who wants some of that? I have some pills on the outside when you guys come out. So this, in my view, is the story of how we go from Mendel, from understanding of the basis of inheritance to these, hypothetical entities that are passed on from generation to generation, ultimately to the embodiment of these entities in chromosomes and DNA, and ultimately to the full mapping of the human genome and all of common genetic variation, to the mapping of the circuitry across all of the different cell types of the human body, and ultimately to the elucidation of the circuits that underlie these associations. This was going to be my 5-minute introduction. I now have a whole talk. So what do we do? It's your call. Who wants to leave and go spend more time with your family? I don't even know what time is it, so let's see. It's exactly 1 hour. So you guys have a choice. I can stop here, take questions. Actually, maybe we should do that. How about stop here, take questions, and then I'll give a quick rundown of the next 70 slides. All right, the floor is open. Your questions are yours. Thank you.
Thank you so much for this beautiful talk.
Thank you. Yes?
Your dominant negative allele was expressed only in adipocytes, or was it expressed in other tissues? Because the mice are smaller from the start, as you know.
Yeah. So the AP3 allele, the particular construct that we're using, the AP2 construct, is known to make smaller mice, but these are even smaller than the small mice to start with. And then the dramatic effect is truly in this part, when you basically see that they're simply unable to gain weight. So basically, when is that allele acting? That allele is acting in the first 3 days of differentiation. After that, it's off. So if you look at whole adipose tissue for a QTL, for a quantitative trait locus effect, you simply don't find any effect there. Yes?
Consider targeting it a bit more than adipose tissues?
So we've basically, we're in partnership with one pharmaceutical company that is now constructing specific targeting, and I will report back when we have results. I feel like you would have enjoyed the second half.
More questions?
Yes?
I think the effect of FTO itself is still controversial because there are these experiments with knockout and also overexpressing mice that show the phenotype.
Yeah, so this is a very beautiful case of how do you dissect the locus between the gene and the switch. Basically, you have this massive switch on the wall that's sitting right under a light bulb. What does knockout do? It just takes out that part of the wall, and it takes out the light bulb too. So basically, when people took out that part of the wall, they took out not just the FTO gene, but also the circuitry that controls IRX3 and IRX5. So one simple way of interpreting these experiments is they took out that enhancer region.
And the overexpression of the protein of the gene, because that was independent of the gene locus.
Well, we can talk about that. Basically, there's a lot of controversial experiments that can be understood in multiple ways. But there are rare coding variants in FTO that have nothing to do with obesity.
Yeah, that is actually where I was aiming.
Yeah. So yes, it's the trouble with genetics. It's very hard to manipulate individual things. If God is listening, you did not do a very good job in separating the functions. Yes?
So I was just wondering today what you would recommend when you're looking at epigenetics. What are the clearest experiments that you find actually end up having an impact?
Are you talking about epigenetics or epigenomics?
I'm talking about whether you would recommend looking at typical epigenetic markers or genome accessibility or something else entirely now.
Yeah.
There's so many techniques
So
available.
if you want to do things at the single cell level, the easiest is single cell attack-seq. So this is the assay for transposase accessible chromatin, and it basically requires very, very little material. We have found in our experience that H3K27 acetylation, so histone 3, lysine 27 acetylation, is the most informative mark of the activity of a region beyond just its accessibility, because accessibility could simply be because a repressor is binding there. Whereas H3K27 acetylation basically tells you that not only is that region, an enhancer, it's an active enhancer. So if I were to choose and I have a lot of cells, I would do H3K27 acetylation. If I have very little material, I would do accessibility. And basically what we do in practice is that for a small number of individuals, we map the enhancers using H3K27 acetylation, And then we go with accessibility and figure out whether changes in accessibility tell us about the differential activity of these enhancer regions.
So sorry, just a practical follow-up. For the ataxic, how many individuals would you normally get the significance of result?
The more the merrier. I mean, you know, it depends on the effect size. I mean, you know, basically, if it's a strong effect, the 3 individuals is plenty. If it's a weak effect, 50 is a good start. And for genome-wide, we usually do 200.
More questions?
Come on, you guys are dying for the second part. Am I allowed, Jerry?
Yeah, I think we better give you medal on the occasion.
Oh, come on, I don't need medals. I just want your attention. Quick show of hands, who wants the second part? 1, 2, 3, 4, okay. Who's done and just wants to go home? All right. So I'll take the medal. Oh, how lovely. It's a Mendel medal. This is very, very pretty. I don't know how many of you. I'll leave it here. You can see it. Yay.
Thank you so much.
This is lovely. That's awesome. So if anybody wants to play with that, I'll just leave it here. Should we do the second half? For those of you feeling tired, let me explain. I started this day yesterday at about 5 a.m. in Boston when my daughter woke me up after 4 hours of sleep. I flew overnight. I landed in Amsterdam, gave a lecture in Vienna, arrived in Brno, and I haven't slept for a long, long time, but I'm actually feeling great. And if you're feeling tired, you can feel free to go home to your family. I'm happy to do another short. Yeah? Okay. All right.
Let's make a 3 minutes break and then we continue.
All right, 3-minute break, and then we're coming back for the second half. And I'll be talking about single cell stuff.